Hello students, in today's class we are going to learn about introduction to prokaryotic transcription. So transcription is basically the process of formation of RNA using DNA as a template. There are three different types of RNAs: messenger RNA that acts as a template for the protein synthesis process, transfer RNA that acts as a carrier of amino acids during protein synthesis, and ribosomal RNA, which form the actual protein synthesizing machinery. So RNA polymerase is the enzyme that is involved in transcription process. It is also known as DNA-dependent RNA polymerase since the enzyme makes use of DNA as a template. During the process, it makes use of ribonucleotides ATP, GTP, UTP, and CTP as building blocks or precursors. It also needs Mg2 plus ions and zinc 2 plus ions as cofactors. The RNA polymerase performs the elongation reaction in 5' to 3' direction using 3' to 5' template DNA strand. So, of the two strands of DNA, there are two strands in the DNA. One is the 5' to 3' strand, and other is the 3' to 5' strand. So, the 3' to 5' strand of DNA acts as a template strand, and the sequence of RNA that is synthesized is basically complementary to the 3' to 5' strand and identical to the other strand, that is the 5' to 3' strand. Hence, this strand is also known as the non-template strand or the coding strand. So RNA polymerase core enzyme is made up of 5 different subunits: 2 alpha, 1 beta prime, and 1 omega subunit. Of these 5 subunits, beta prime subunit is the main catalytic subunit. The beta subunit binds DNTPs and also interact with the sigma subunit, which is the 6th subunit of RNA polymerase. The alpha subunits appear to be essential for assembly and for activation of the enzyme by regulatory proteins. And the omega subunit is important for assembly of RNA polymerase and its stabilization. RNA polymerase holoenzyme basically consists of the 5 subunits of the core RNA polymerase plus 6 subunit, which is known as the sigma subunit. There are different variants to the sigma subunit, which differ from each other by their size. Sigma subunit is only transiently associated with the core RNA polymerase, and it directs the enzyme in identifying which sequences to be polymerized or transcribed or where specifically bind to the DNA. These 6 subunits together form the RNA polymerase holoenzyme, also known as RNA polymerase complete enzyme. In case of E. coli, the sigma 70 is the most common sigma subunit, which is involved in expression of the most genes in E. coli. While sigma 32 is one of the variant that is involved in expression of a group of genes involved in a heat shock response. Coming to the error rate of the transcription process, during the transcription process, there is approximately one error every 10 to the power 4 to 10 to the power 5 bases that are being synthesized. This means that there are quite good enough number of errors if we compare the process with the replication process. This is because the enzyme involved in DNA replication, that is DNA polymerase, has additional activity which is known as a proofreading activity. So if any wrong base is being added during replication process, by the enzyme, then it cleaves off or removes the wrong base by an exonuclease activity, which is known as 3' to 5' exonuclease activity, that cleaves off the wrongly added base by identifying the mistake. This activity, which is known as proofreading function, is absent in case of RNA polymerase enzyme, and hence it makes comparatively more mistakes as compared to the DNA polymerase that is involved in DNA replication process. So approximately we can have one mistake or error for every 10 to the power 4 to 10 to the power 5 ribonucleotides that are incorporated into RNA. The next slide talks about the promoter elements. So promoters are basically regions that are being identified or recognized by the RNA polymerase holoenzyme, and it begins transcription at the sequences immediately adjacent to the promoter regions. The promoter regions in case of prokaryote generally extend from minus 30 to plus 30 bases in position. So the transcription start site is basically denoted by +1 position, while all the bases that are present before the transcription start site are given a negative denomination, and all the bases after the transcription start site are given a positive denomination. Two types of sequences are important in case of prokaryotic transcription initiation. So these promoter elements include -10 region, which has a sequence that reads stat at in 5' to 3' direction, and another conserved or consensus region is at -35 position, which means it is 35 base pairs before the transcription start site, and it reads TTGACA in 5' to 3' direction. So how are these prokaryotic promoters identified? So basically different genes, rather upstream sequences of the genes are being copied, and we make use of a program which is we make use of a program such as cluster W or cluster omega for multiple sequence alignment, wherein after alignment of all these different sequences, what was being found that some regions were conserved um, or similar in all these different sequences that were being used. And one of the sequence that was identified was the TAT-AT region or the TAT-AT sequence that is present at the -10 region, and TTGACA region that is present, TTGACA sequence that is present in the -35 region. So that's it about introduction to prokaryotic transcription. Thank you.